Okay. The handout, this is an important piece of information, this document here that has our playground, okay? It's kind of like, for me, one of the big objectives of this class is for you guys to be able to just have a general sense about animals and how different groups are related to each other. And to me, basically most of the information that you'll need to know will fit onto this playground by the end of the semester, okay? Because think about what this, all of the information that we would put on this tells us who's related to who, who's old, who's new. It tells us, once we start putting all the characters on here, it tells us why groups are related to each other. It tells us which are the oldest animals, so on and so forth. So we're going to keep revisiting this. And I think that you probably know more about this already than you suspect, right? So if we just kind of look at this and bring in what we've already discussed. All right, what's the first group? And again, we think about this as like a time aspect. If we go further back in time, what's our oldest animal ancestor? Our living animal that is, we share most, the ancient common ancestor would be the sponges. On the peripheral. Okay? And, you know, we could put a bunch of, we don't really talk about these characters down here that unite all the animals, right? But there are things that animals have that make us monophyletic groups and make us a group and separate us from things like fungi and other protists, right? Okay, so the first group would be the sponges, and then the next group here, and this is one of the things here is multicellular, right? So all animals are multicellular, all animals undergo development, all animals lack a cell wall, right? We're not going to really get into these characteristics, but so sponges are the first group of animals, and then what would our next one? It's right here, the Cnidarians, right? And then we get into these groups here. 1, 2, 3, and then 2, and then this guy over here. Now, what is our character between the sponges and the Cnidarians that sort of separate those two? And really, we can put two things on here that separate sponges from all the rest of the animals. In a lot of ways, it's kind of like sponges and all the rest of the animals, right? So what are two things that sponges lack that all of the animals have?
Sponges are asymmetrical, while the Cnidarians have a radial symmetry.
Good. And symmetry with two M's? Symmetry. I'm going to try that. And then they also, after the advent of multicellular sponges, we get the advent of symmetry, and we also get tissues. Okay? So sponges are multicellular, but barely so, right? They're made up of multiple cells that do work together, but very loosely, right? Here we get into the development of true tissue, specialization of those cells. I do this, you do that, right? We're going to have really specific tissue types. Symmetry, again, is symmetry related to genetics? What do you think? Is symmetry a genetic trait? We don't talk about development a lot, but is whether you're radial symmetrical or you have symmetry at all, is that determined by your DNA? Yeah, absolutely, right? Because remember, You start life, sperm plus egg equals a little baby, whatever, sponge, jellyfish, all the way up to us. How those cells divide, and which ones divide more, and which ones die through apoptosis, and how you develop is determined by the DNA, right? So sponges don't have the genes to be symmetrical. Here we have symmetry genes, okay? And the Nidarians specifically develop using radial symmetry. Okay. And then from here out, so we have symmetry there, and then one branch goes to radial symmetry, the other branch goes to bilateral symmetry. Okay. And then we get this main branch. This branch, this is protostomic development. And here we have deuterostomic development. And this one, I'm less interested in the adaptive function of that. I mean, we can talk about the adaptive function of symmetry, right? If you're radially symmetrical, you're just kind of floating around, dealing with everything all around you. If you're bilaterally symmetrical, it means you're moving in one direction and you're concentrating everything into a head end, right? Deuterostomic, protostomic, so again, baseline development. Sperm plus egg equals the one single cell zygote. But then it divides to 2 cells, and 4 cells, and 8 cells. And then you get into this ball called the black tumor, a bunch of cells. Mitosis. Right? And then that ball gets a little dimple, and that little dimple continues to grow, right? Until you end up with basically would be like a donut. If you're looking at it on the end, if you're looking at it from the side, that's the very simplest possible body plan. A tube within a tube. Okay? So if we take this and we push our finger in there and we push a tube all the way through, that's what happens during development. Okay? And then what is the tube? What is this thing here? That's our mouth and our anus. That is our GI tube. That's the basic body plan of almost every animal, right? It's just a GI tract that goes through the body. Here's the rest of the body. This might develop later into the legs. You might develop a backbone, right? There's all different ways of modifying this real simple plan, but that's the basic body plan of animals, right? Now, in a protostomically developing organism, and that would include, this includes all of these. Any animal that's on this branch that includes all of these phyla, i.e. flatworms, mollusks, segmented worms, roundworms, and arthropods are all protostomes, right? Because they're on this branch. In that group, what makes them a group is that the first dimple that starts to form, the first opening, here's opening, here's no opening, the first opening becomes the mouth. The pre or pro means like first, the stome is mouth. So the first the first opening develops into the mouth. Here, the stone is second. So in a protostome or deuterostome, the first opening that develops is the anus. So it's just, but again, I don't really know why I'm belaboring this because who the hell cares? It's not like that means anything. It just is what it is. right? That just, so a lot of times you find a new animal, well, not a lot of times, but they have found animals and they don't know where they go in this tree. So what they'll do is they'll go to very early stages of development and keep track. There's this first opening, the anus or the mouth, and then that allows them to place them either in this part or this part of the tree. Okay, it's just a characteristic that is used to place animals on this tree. Okay, protostamic development, we can break these into two main clades here. We have this clade, and we have this clade. We have this clade, and we have this clade. Okay? These animals, we think back to the annelid or the mollusk lecture, and we talked about these funky, weird little larvae, really early stages of development. And they have these little cilia on them, that's how they move. Tiny little stage of the life cycle. And during that stage, they have cilia, and that the presence of this part of their life cycle, so it would go something like egg to a ciliated larvae to a juvenile to an adult, right? If you have this stage of the life cycle, then you are put into this group here. That includes the platyhelminths, the mollusks, and the annelids. Okay? So that's what unites this main group of protostomes. Presence of ciliated larvae is the character that makes this a group. Okay. Now today we find ourselves up to this point, right? So by the time midterm is done, we're going to be talking about all of the invertebrates for the class. Right? There's the good news. Again, I'm trying to come with an optimistic, positive message this morning. The other half of the semester is just this piece of cake, right? First half of the semester, we've covered all of these phyla. Second half of the semester, we're just talking about chordates and echinoderms. Okay, so I'm hoping maybe that's what you need to hear this morning. Keep going. Okay. Now, this group we're referring to. So these are the this clade. So keep in mind, these are the traits. These are the groups. Okay? These are the traits that define the groups. But these are the groups. Or clades, we call them. Low, animals, zoans. These are the ecdysozoans. So the trait that unites them is they shed exoskeleton or cuticle / cuticle. So animals in this group, including the nematodes, the arthropods, and the tardigrades, all share a characteristic that they have a hard exoskeleton, and periodically they shed that. Okay? So that is the character that puts them into this clade, ecdysozoans. So nematoda and arthropoda. And then this clade over here is the deuterostomes. Okay. That includes us and the chordates. and the spiny skin, echinoderm. Okay. That's about the level of detail we're going to go into on this clay frame. Okay. Yeah.
Can you repeat the name of the two plays?
Yeah, absolutely. These two?
Yeah.
Okay. So I'm going to write them in big letters here. So that low zones zoology, right? Local tropozoans, these include, so this is the presence, it's a group of animals. I'm going to call it, we'll use the nerd word, a clade of animals with a ciliated larval stage. And the other group is the ecdyso animals. And they are a group of animals clade that sheds outer layer or something like that. And I mean, you can look at other things that characterize them. If you look at their DNA, they'll show that they're more closely related to. This again, for me, is just a logical way. This is how zoologists sort of take a million species and divide them into piles, right? Like the tardigrades. We're not going to really have a lecture on them. They're not going to be on the lecture exams. You guys have that one assignment where you're going to take some pictures of them. But these things, you may hear about them. They're very cool animals. And this tells us that you can predict some things about them based on where they fall out on this tree, right? Because you can say, well, they must be protostomes, and they must shed their exoskeleton, right? Because of where they occur on here. This gives you a framework for thinking about animals. Other questions about this cladogram? This is one of the, honestly, this is the only cumulative thing that will be on the final. There's some version of this information. Okay.
Does there need to be like organ and then like organ system on there, or are we just?
No, yeah, because I did ask about that on the last test, right?
Yeah.
I know, nobody's ever asked me that before. I want to be fair in that, on the final, no. I may, on the next test, as you're studying, you may be learning other little factoids about arthropods that you should be able to place in the context of this, right? But as the final large cladogram will have this level of detail, right? But that's where the higher order thinking comes in, is sort of making those connections, right? To say, for example, if I asked you where you find parasitic organisms, you'd say, really just up here. If I asked you where you find predators, so you should be able to kind of combine the two in a way. I don't know if I'm making any sense at all on that. Okay. We'll keep reviewing this, and we'll keep touching base with this. And in fact, we probably will expand, because we're spending basically half a semester on this. So what kind of animals do we find in this group? This is the group of animals where everyone comes into class thinking we're only gonna be talking about panda bears and snakes and sea turtles and things. Pretty much all of your favorite animals probably are right here in this group. Right? But most of the diversity of animals is all out in here. Right? And in fact, most of the diversity, this is a perfect segue. I totally did not plan this, bro. Right here. If you just look at break down the number of species in the animal kingdom, most animals are arthropods. Most animals are either spiders, crayfish, particularly things like beetles. Arthropods are just one phyla up here, but in fact they're much more important than just that. Think about the ways they impact human life. All the diseases that are carried by things like mosquitoes. Think about all of the pests that affect our agriculture. Think about food sources, right? Everything up here affects humans. You guys know, what is this one here?
Horseshoe crab.
Horseshoe crab. You know why one of the reasons horseshoe crabs are really important for humans? This is something this still blows my mind.
Don't they use their own blood for cancer treatment?
They use their blood to test for the type of bacteria. And it changes color in the presence of a certain type of bacteria. And it's so incredibly sensitive that they've not figured out a way in the laboratory to test for this bacteria without using the blood. So they harvest thousands of these things every year, put them in these strange apparatus, draw their blood, and then use the blood in these tests, right?
And
Narwhal also
narwhal
a
is
different color too.
No, this is the, this is cool for like new teachers. Peppermint narwhal, this is the group that, or I think it's probably a person that does all these informatics. Tons of really neat informational graphics like that. You can check. Really, really neat stuff. I love the, look at this. Do you know that there's how many species of giraffes? I mean, honestly, this sounds stupid, but I learned just doing stuff like this, that there are 1, 2, 3, 4, 5, 6, 7 species of giraffes. I thought there was just one. Right? Pretty interesting. But anyways, peppermint narwhal, check them out. Oh, here it is. Okay. Talk about this. Okay. So we are going to kind of approach this from the level of the subphylum. Okay, so these are the major subphyla of arthropods. Okay, and here's some of the overlap between the lecture and lab. Right. Today we're going to talk about these first 3 subphylums, and the biggest one we'll talk about on Thursday, which is the hexapods, or the 6-legged insects. Okay. This level of detail, don't worry, this is just kind of a framework for us as we approach this lecture. You don't need to know all the details in this by watching. Great book called Diversity of Life. If you're really a student of biology and biodiversity, it's a good one to read. And this informatics, this little diagram here, it shows if you take all of the major groups of living things and projected them proportional to how many species there are, this is what it would look like. Okay. So all of the fungi species, known described species of fungi, would be this big. Here's all the plants. All right. This one surprised me too. But again, this is not to say that there are not more species of plants, but that this is how they have been formally described. And so what's the big story here? In terms of, if you want to get a real quick visual impression about where the diversity of life lies,
And the hexapoda is the biggest.
Yeah. These are just the insects. Here's the other arthropods, right? What are we all, what's the real sexy groups here? Mammals, birds, maybe reptiles, if you're really weird, amphibians, if you're like some kind of sicko. But in reality, these things are dwarfed by the diversity. If you want to study a group of animals and make discovery after discovery after discovery, forget about these things, right? It's right there. This is a very interesting question that comes up is why is this so successful about this design that has created so many species, right? So be sure to check that out. What are the things that unite this, the shared derived characters of arthropods? The big one is these paired jointed appendages, right? So in other words, they have one on each side, and they have, look at all the rest, they have joints. Other groups, even phyla up to this point that have appendages at all, really there aren't. I mean, some of the annelids have little legs and things, or pseudopods. Some of the mollusks have little tentacles, but nothing like arthropods. Right? These arthropods, and included in these appendages are things like mouth parts, wings, legs, swimmerets, all those things that we look at in lab. Those are all grouped into this, this thing called appendages. Okay? So many of those things become really specialized, which allows, this goes back to this question of why are there so many species? This is a very adaptable group. They can use these appendages to exploit so many different resources, right? They can be predators, they can be herbivores, they can be filter feeders, they can be, you name it. If they specialize those appendages, they can do it, right? The exoskeleton is made of this material here. And you may remember back in 120, if you really have a good memory, this is a carbohydrate. It is a structured carbohydrate similar to cellulose that's found in plants, right? And that's what their skeleton is made out of. It is an outside skeleton, which they molt through ecdysis, right? They shed that exoskeleton periodically as they grow through this process of ecdysis.
You said it's made on a plant?
No, so this material, chitin, is similar to the material that plants are made out of.
Okay, thank you.
So, ecdysis, this is the different steps in ecdysis, right? So can you hear the essay question? Please describe the steps in ecdysis, right? This is the kind of, when you're going through and looking at these presentations, if you see steps, you see comparison, those are the things you really want to look for on the test. So you guys seen Tommy Boy? That guy in the little coat thing, do you remember? This is what we're talking about here. These animals grow to a certain point where they can't grow anymore because they have this inflexible, non-living exoskeleton around them, okay? So their growth becomes limited by that exoskeleton. So in order to get bigger, they need to shed that exoskeleton. And that's this ecdysis, okay? So this is the steps. Oh, another couple of points about ecdysis. This term is a good one to know, instars. So when we have, here's a baby, we'll just there's a joint that attaches. There's a first instar. And then here's a little bit bigger one. And then here's the big one. The growth, you've got to change the way you think about arthropods because there is no really in-between size, right? They grow to this size, then they can't grow anymore because of the exoskeleton. So they have to drop that exoskeleton so they shed, grow quickly, and then harden up at a larger size. So these stages between molts are called instars. Right.
So in stores where they drop or where they shed?
It is in between shedding, I guess you could think. And so when you're kind of keeping track of an insect's life cycle, then you refer to those periods. They grow, they're growing underneath there, they shed. So they get to a certain size, too big. The inner layer of the exoskeleton is reabsorbed. So now you have basically a space between the epidermis, this is the living tissue, right? And there's a space between that and that non-living exoskeleton. Okay? Why is there a space? Because that layer in between has been reabsorbed. Okay? Then they start to secrete a new exoskeleton over the epidermis. They hulk out and break out of the old exoskeleton. Okay? At this point, after they've broken out of the old exoskeleton, they still have a new but soft exoskeleton. Okay? They grow as quickly as they can, they expand before this soft exoskeleton hardens. And then once it hardens, that's it. That's as big as they're going to get for a while. Okay. So this is lots goes on during this process. And that is how they're allowed to grow. Because they have to get rid of that old hard skeleton. Until the new exoskeleton hardens, this is what a soft shell crab is, right? A soft shell crab is at this point in this cycle. Right? The hard exoskeleton has not formed. So we can kind of this video here. So this, didn't know, well this is cicada, it says right there, but these guys, this is the larval stage, or juvenile stage, it lives underground, sucking on the sap of tree roots. They then crawl up onto a tree. At this point, he's probably reabsorbed the layer between his living and the non-living outside. And he swells, she or he. So it breaks through, climbs out, and at this point, this is really weird. The wings are kind of like they are really they're soft and pliable, and they're hollow. They have a bunch of like hollow vessels, kind of like an inflatable air mattress or something. And now the cicada starts to pump lymph, like blood and things out into the wings and blows them up, expands them. Okay, and while they're soft like that, they have to do everything they can to get bigger before that new exoskeleton starts to harden up for them. Every type of arthropod goes through this. Spiders, beetles, flies, mantids. So on and so forth. Right? So that, crayfish, there's the old exoskeleton, and we have this little soft guy underneath it. And expand and harden. Right? You pay attention, yeah, come on.
Is it like a binary occurrence, or is it like a once in a lifetime sort of thing?
It depends on how long the arthropod lives, but they certainly, they all, I would guess they all molt more than once. And it depends on how rapidly they grow. People think, and I know this sounds like maybe a silly question, but are arthropods and snakes related? No, but it's a similar kind of analogous type of a situation where, but I think snakes shed for different reasons too. Shed parasites and things. So, but they'll shed multiple times. As a group, the arthropods also have open circulatory system. Okay, so we compare that with a closed circulatory system. So here's the body, and here's the heart. In a closed circulatory system. And again, this is just a universal challenge that animals have. They need to distribute. They need to distribute the nutrients, they need to distribute oxygen, they need to distribute stuff. How do they do it? Usually in the blood, right? So in a closed circulatory system, the blood leaves the heart and artery and returns back to the heart in a vein, and in between they have these little capillaries. Right. The blood is always in a vessel. The blood is always inside of either a vein, a venular artery, anything like that. Okay. It's always closed into a system. In an open circulatory system, the blood leaves the heart. I should have drawn this up there. The blood leaves the heart into just a general cavity. And sometimes it's called a hemocoel. Right? We know heme means what? What's a hematologist? Blood. A blood seals. So it would be HEMOCOEL. Blood cavity. Hemocoel. Okay, and then it sloshes around, not enclosed in anything. It's just in there, bathing the tissue in nutritive blood, and then it somehow finds its way back into the vessel and makes it back to the heart. Right. That describes all arthropods. Does not seem like a very effective system to me. Right? It does not seem like an effective system. However, you can't argue with results. Right? Think back to that diagram of the diversity of life. Right? It must be working, because 1 million arthropods can't be wrong. Right? Mostly there are male and female arthropods, so their sexes are separate. Mostly are oviparous. Anyone want to guess what this term means? Oviparous. Another very good term. Vocab term. I.e., it's a great film. Oviparous. Right? Ovi, what do you think of when you hear ovi?
egg.
Egg. How do they give life? How do they give parity? They lay eggs. So they're egg layers. Okay, we are viviparous. We give life birth. Animals are viviparous. And then there's this ovoviviparous. It's kind of like a mix of the two. But they mostly lay eggs. They have separate sexes. Well-developed hearing, eyesight. Check out the mantid shrimp. If you want to see some really freaky animal, the mantid shrimp can see like in colors that we don't even know exist, right? They're so fast they have these little jointed appendages that they're like this big and they can crack through aquarium glass. They're that fast, right? Hearing, chemosensory abilities, very good sensory structures. Incredibly diverse, abundant, found everywhere from the deepest ocean to the Arctic, tropics. High degree of endemism. Endemism means if you're only found in one specific place up on Earth, then you are endemic. Okay. So an example of that would be there are crayfish that are only found in the Etowah River. There's a type of some kind of arthropod that is only found. If you guys have been to the top of Stone Mountain, it's really pretty. You can forget about all the politics and everything. When you're up on the top of this granitic dome looking out over Georgia, it's really pretty up there. But there's these rain pools that form, and there's a type of arthropod that's found nowhere else except in those little rain pools on the top of Stone Mountain. Right? Highly endemic. Very cool.
What word is it again?
I'm sorry?
What word did you say means?
Endemism.
Thank you.
Yep. So, and again, this diversity shouldn't surprise you that they're found, right? And you find little temporary pools, dry, wet, cold, hot, ocean, fresh water. Yes. Arthropods are everywhere. They are everywhere. So, and this is just, again, a real quick tour of their diversity. Body form, sizes, shapes. That's the thing that lives on, like, you guys know sea monkeys? You can buy the eggs and hatch them out of brine shrimp. So
What's this word? Sea monkeys are?
they're a type of, that's a brine shrimp. That you can buy the eggs and hatch them out, and they're pretty weird little arthropod animals. It used to be like you could buy them in the back of magazines and stuff. I don't think people read magazines anymore. You guys know what a magazine is? No, just kidding. So we're gonna start out with this first subtopic, the Mad Magazine. And Myriad Poet. How are you gonna remember this as a name? Somebody give me a way to, what do you find in the subtitle Myriad Poet?
Centipede, millipede,
Okay, so connect that to the name.
many legs,
Yeah, myriad
feet, and feet.
toads, myriad of many. So these are the many-legged animals. See, they're pretty basal to the rest of the arthropods. Good comparison here. And this just means they're easy to draw, many undifferentiated segments. Or make a video, there's video games about this too. It's just a bunch of that, it's just easy, right? How do you draw a centipede? Undifferentiated segments. The segments are all the same, and then just put some legs on them, and you got a centipede. Looks like what?
A caterpillar.
Yes, but a caterpillar would have an antenna. So,
Oh.
yeah, that's the difference. Don't challenge my zoological
Well, it
knowledge.
says single pair of antenna.
Oh, okay, well, never mind, I didn't say that. Okay, here's the difference. This is really serious now. This is getting serious. On caterpillars, there's actually only three pairs of true legs. and these are pseudopods. On a centipede, all the legs are the same on the way to the front and the back. So, okay, I can move on now. Centipedes are predators, but many of them are venomous. They're very active, and they don't have a 100 legs. These guys don't have 1,000 legs. Centipedes do have fewer legs because they have one pair of legs per segment. Okay. Millipedes have 2 pairs of legs per segment. They are a little bit less active, and they are kind of peaceable vegetarians or detritivores. Okay. So these ones are kind of scary. These ones are not. Oh, these guys can spray cyanide out their butts, so not to be trifled with. Centipedes and millipedes. So here's that millipede. See how he has 2 pairs of legs per segment. Different types of centipedes. Okay, that's all I really want you to know about myriapoda. Okay, know that they're in that group. Know the difference between the centipede and millipede. Okay, that may be something you will remember in 10 years. Right. That's my goal. We move into this subphylum, Chelicerata. Okay, they have these first pair of appendages are formed into these things called chelicerae. Okay. If we look at the head of different arthropods, these are those first pair of appendages. They're formed into, basically, in spiders, these are fangs. They are usually hypodermic in that they're hollow and they can inject stuff, right? Venom. So members of this subphylum have mouth parts like this rather than like this. Okay. Many arthropods have mandibulate mouth parts, not the. This includes the most famous groups are the spiders, but we're going to see all the arthropod or the what's the spider is the
Arachnid?
arachnids. Thank you. Ticks, chiggers, all that are found in this group too, and then the horseshoe crabs. Okay. Here are those appendages, hook-like fangs for injecting digestives and enzymes. And then 4 pairs of walking legs, except in the horseshoe crabs. So 8 total legs in most of the Chelicerata, compared to 6 for, or 3 pairs for the hexapods. Yeah. Chelicerata. So 2 classes that we'll talk about. We have, so I would recommend keeping yourself, for each test, certainly, keep yourself a list of the groups that we're discussing here. So we have, right now we're in phylum. What phylum are we talking about?
Arthropoda.
Arthropoda, Arthropoda. The subphylum, talking about Chelicerata, and then we have kingdom, phylum, class, marrow, so long term. This is the horseshoe crabs. Maybe you guys can read, this is available to you. This is the kind of stuff that you just memorize. I don't really need to explain it. This is just like the factoids about each group. There's only 5 species, but they're very unique in the arthropods. They're probably an ancient group. They share a lot of really primitive similarities. This is just a body part. Where do you suppose this body part stretches from? The
Head to the floor.
So that's basically like a big shell that covers most of their body. Not the abdominal, but thorax and the head portion. They get oxygen through these things called book gills. If you happen to book a beach vacation during their reproductive season, it is not very pleasant. These things are not something you really, swimming with dolphins is one thing, but a bunch of creepy horseshoe crabs is not particularly pleasant. Really common. I think this may be the only genus of for these 5 species, living horseshoe crabs. That would scare me as a kid. Look at that. They look like dinosaur things. Flip them over. Here's that cephalothorax. There's the abdomen. The book gills here. Here's all the little 5 pairs of appendages. And then the class Arachnida, also Chelicerate. So they're grouped under the same subphylum. Lots of species. And I'm really excited. This is one of the things I'm excited about. The violets is this lady, Rebecca Godwin, is coming, and she is an expert in spiders. So I'm excited to learn. I'm still a little afraid of spiders, kind of like a little child. Centipedes and spiders still, but here we have access to a world-renowned expert. She's a badass spider person, so we're gonna get out in the field and learn about spiders with her. Harvest
Maybe
Minute.
someone else is coming too.
Huh?
Is there anyone else coming?
Oh, there's a whole bunch of really great scientists coming, yeah. Harvestmen, these are daddy longlegs. They're not spiders, but they are arachnids. They're not the most venomous animal on Earth either. But many venomous groups found in this arachnida. Okay. Which one of these are parasitic? Ticks. And chiggers, ticks. They obtain oxygen through these things called book lungs. They remove nitrogenous waste using these structures, mouth-piggy tubules. So our analogous organ would be a kidney. So this is like a kidney of an arachnid, the mouth-piggy tubule.
Borrow, a bank for all of us.
This is called a bull spider. I think they live in Australia. This is the female, I believe.
Active approach to catching prey, dangling a silken thread from her outstretched leg. A familiar flail or a bolus. The head of her weapon, in lieu of a lead ball, is a blob of sticky thread.
Okay, so her genetics allow her to make this protein. She has an organ called a spinneret. She spins this, makes this protein, and it is spun into silk. That silk is the same as silk as made by a bunch of other arthropods. Scientists cannot recreate this material. The strength of this silk, the flexibility of this silk, you just cannot go into a lab and make this. You need a spider to be able to recreate this material. Yeah, Samantha.
So saying it's silk, is that the same type of silk we use for?
That comes from a caterpillar. But it's a very, very similar type of material, yeah.
Okay, yeah, I was curious about
To lure a male moth into range, the bowler spider cleverly mimics the smell or pheromones of a female moth. Swooning into range, the male is struck with the bolus. The spider reels in its prey and delivers a deadly bite.
So how is how is it that this spider also has the DNA to produce pheromones that are produced by moths? They produce the pheromone lures in the moth, and then the spider has then created this little weapon
The
that spins
Bachelor
around.
is back. Stream it live on ABC.
It sticks this to the prey. I don't know. That, to me, this is all from natural selection, right? Somehow this has been created. Right? It's used to capture food, also make shelter, bridges, reproduction. They wrap their eggs in. You've seen those little spider egg sacs, right? The other thing that spiders do with this one also blew my mind. They do this behavior, it's called ballooning. So the spider crawls up to the top of the camera here and starts to let out a little bit of silk, a little bit more silk, starts to be captured by the wind. And at some point, the silk is long enough that the wind picks up the arthropod and takes it up into the atmosphere. And at any one time, there are millions of arthropods just floating around in the atmosphere on these balloons, right? So they can just disperse anywhere they want to go. I just think that's too darn cool. Silk spinning. These trapdoor spiders. There's one that I know that Rebecca's going to be interested in looking for. It's called a purse spider that we have at Dr. Flint's. They produce these big purses on the side of trees. Funnel spiders, all kinds of unique. Silk spinning. Fish spider, these guys have figured out how to distribute their weight lightly enough that they don't overcome the surface tension of water and they can just walk around on the water. These are real common plants too. Good mothers, they carry their babies around on the back. So spiders are very, you like to reduce them to the underside of our shoe, but in fairness, they're a very unique group of animals. The two that we have here that are dangerous are the brown recluse. You can see they're called brown fiddlers. They sometimes have this little mark that looks like a fiddle on their thoracic segments, on black widows too. These are both common spiders here. I know people that have been bitten by both of them. Scorpions. We do have scorpions here too. Ticks, chiggers, harvestmen. And then the final submodel here, the crustacean. Okay, and I'm going to go. I can kind of feel the energy in this room is pretty much shot to hell. I'm going to go through this pretty quickly. All this is up on canvas. I would focus on this one particularly, looking at this. Let's see, we have subfinal crustacea. Focus on the taxonomy here, because there's several classes, and I think even down to the order level. Okay, crustacea are mostly aquatic. Okay. Think about these crayfish, shrimp, amphipods, things like lobsters, crabs. Mostly these are found in the water. There are a couple of terrestrial groups, but they're mainly aquatic. They mostly are detritivores. Okay? We don't see a lot of predators in this group. These things are mostly just eating dead, decomposing stuff. Okay. As a rule of thumb about this group, about this subphylum, there's a couple of them. Other things that unite them as a subphylum, as a monophyletic clade, they have this calcium carbonate exoskeleton. So they do, because they're arthropods, they have chitin, but added into that to make us a harder shell, they have a calcified exoskeleton. 2 pairs of antennae, this is more trivial trivia. 3 pairs of feeding appendages, head and thorax fused into the cephalothorax, very similar to the arachnids, and in fact similar to the horseshoe crab. Right. So the most important thing I'd say on this slide is this first one. I'm not going to ask this level of detail about, subphylum. Classic crustacean here. So the head and thoracic segments are fused into this cephalothorax. But lots of different pairs jointed appendages adapted and specialized for different structures, for different functions. Okay. This is just right out of the lab, looking at swim lanes, uropods, walking legs, right?
Byron.
Okay, yeah, I think there's even a diagram on this next one. So biramus means that they have 2 different segments, 2 different branches. They're branched. Their appendages tend to be biramus or branched.
RS means what again?
Yeah, so this word back here, biramus appendages, means that the appendages are often branched. So again, I'm unclear on what the functional significance of this is. This is more just another taxonomic trait used to classify these groups. The class, now the strata, so the same level of classification as the arachnoid, etc. Class, called strata, and we have order, Arthropoda. This includes this is all the same name for, are different names for the same group. Willipoles, pillows, they are all of the same group. They're found in the order Arthropoda. Dorsal ventrally flattened means in the dorsal and ventral plane, they're flattened. So this is like a pancake. You can find them, you can find roly-polies and pillbugs and all these in the ocean, terrestrial settings, caves, so on and so forth. They will brood their eggs. The female will brood the eggs at the base of the leg, so she actually carries the fertilized eggs around with her. And these things called marsupial. Here's some different isopods, different species, or decapoda in the class, not or decapoda. And this includes shrimps, lobsters, crabs, but in this order. Yeah. It's a big one in terms of their importance to humans. Right. Lots of good food here. lobsters, again, these things very long lifespans, should live up to 20 years or more. Crayfishes, we'll look at crayfishes in the bottom list. There's another person coming that knows a lot about crayfishes. Yeah. These are all cephalopods. Right, so this is the weird, crabs actually have a little tail, just like a lobster, except that it's curled up underneath. So here's the eggs of the female. You'll scoop them back there and curl these containers. So, I know that's a lot. The last, especially that last portion here, this mouth distraction, I think if you kind of understand that this order contains those organisms, these ones include the bugs. I'm not going to ask a lot of detailed questions about there's a lot of thoughts on it. But just focus on how they all fit together in this subfile, the three subfiles we talked about. All right, I'll see some of you guys in just a few short minutes.